Hi everyone, we've got a full house. It looks great. I see a lot of my students out here, a lot of my former students, so it's so great to see you all again, and thank you for joining me on your Wednesday afternoon before finals. I am going to be nursing this water throughout the talk. I've got a little bit of a cold going on, so my voice is kind of going in and out, so if you can't hear me in the back, let me know and I can try to speak a little bit louder. But I am excited to be invited to talk to you all today about my research. So I'm going to kind of just do an oversight of some of the work that I've done in the past and what I'm looking to do here, what I'm continuing to do here at ETSU, and hopefully teach you guys a little bit about how we can study babies, because it's something that I think is really unique, and I love to just tell people about how we can learn more and more about how these really young infants can tell us about how they're learning about the world. And they're able to do so much more than sometimes we give them credit for, and we just have to figure out a way to learn what they're trying to tell us. Today I'm going to kind of work through several different objectives. I'm going to start by introducing developmental science in general, and then walk you through a little bit more about what social perception is. Okay, and then we'll move into several infant competencies that we've discovered within the last couple of years about what infants are able to do when it comes to social perception. So what do they know about faces? What do they know about bodies? And then within that together, looking at what do they know about facial emotion, body emotion, how can they process and understand different things that they're seeing from their world. And then finally, I'll end with a brief discussion on some of my newer research that's comparing cognitive development in opiate-exposed and non-opiate-exposed newborn infants. So broadly starting with a discussion on what is social perception. It could be a term that maybe you guys have heard before, but we don't know exactly how we're going to define that. So for me, social perception is just how we process information about, how we start to make inferences, and how do we remember different people. So imagine you're walking down a busy street, much like one on this image here on the screen. How do we understand what the environment is that we're in? How are we able to identify these different individuals from our environment? Can we recognize them from a distance? Can we recognize them when they're facing us? Or what about when they're not facing us? What does that process look like? And social perception, or this kind of recognition of different individuals, helps us understand that a little bit better. Social perception can help us understand whether we need to run towards someone, or maybe we need to run away. So this identification of different emotions and what their facial expressions are showing can help us succeed in our environment, right? To help us stay safe. So our ability, our as an adult, to accurately and quickly identify these various components of social stimuli, so social people, so faces, bodies, their body posture, emotions, right? That's going to help us respond and react appropriately in these different situations. So being able to extract things like emotion, gender, race, identity, that's what we're talking about when we're referring to social perception. Within social perception, I'm going to focus a lot of the topics today on spatial perception within social stimuli. So spatial perception is really cool because it's a way that, or it works closely together with social perception because it helps us process faces. And you may say, how is it doing that? Spatial processing is completely different from social processing. Well, in reality, if you think back to all of your classmates from maybe high school, okay, you're looking through that yearbook. Just by looking at their faces, you can probably still, without looking at their names, you could identify a lot of those different individuals. Okay, they all have different faces. You've got a memory for those people, right? But one of the ways that we're able to differentiate between all the different people, right? I'm looking out in the audience today and I can tell that all you guys are different individuals, right? And one of the ways that I can do that is by looking at the specific featural information within your face. So everybody has got two eyes, above a nose, above a mouth. Okay, all that's the same. It's not going to vary from person to person. But what really helps us do this identification is understanding the differences between those facial features. So looking at the specific spatial relations. So what is the distance between the eyes or the distance between the nose and the mouth? So looking at the spatial relations among facial features. And so when we're able to look at some spatial information like that, that's something that we do as adults without really having to think about it. So in infants, we're trying to understand, when are they able to do what we do as adults? When do they learn this? How do they learn this? And so by examining spatial processing in social stimuli, we can see when that skill develops in infants and know that it's got this really importance for later on our success kind of in the world, being able to identify different people, being able to recognize emotions, so on and so forth. So we want to be able to, there's this theory out there that it's one's experience with different social stimuli, one's experience with faces that we gain over the first, so many months, years of life, that helps us become experts, that helps us be able to identify all these different people for one another. Okay, so there's that theory of expertise that's involved here. And so what one of the goals we're setting forth to do is understand how that works with faces. Okay, how do we become expert face processors? So I alluded to this a little bit, and I think this picture will help kind of you understand a little bit more of what I mean by the spatial relations across features. So when we're talking about experience that we've gained over time with faces, why do we think social stimuli are so special? Well, there is a model of face processing that shows that our expertise arises from the fact that we can process faces as a whole rather than as their individual component parts. Okay, so that's, maybe you've heard this in some of your classes before, but it's holistic processing, right? The sum is greater, the whole is greater than the sum of its parts. There we go, right? So kind of along those lines, looking at the whole face as one object rather than a collection of individual features. And that's part of expertise processing too. So learning if we do that, we don't really need to pay attention to the individual features. Do I have a laser on here? But instead we're really looking to the distance between the eyes and the nose and the mouth there to make these sort of judgments. And so I say we do this for faces, well, we do it for bodies as well. So bodies are another source of social stimuli that have a lot of similar features to faces. Okay, so they're organized in a similar fashion, or we could impose that same organization if we tried. They have a face or a head on top of a torso. It's got these two limbs, right, extruding from it, and then two limbs down below. Okay, so it's got a similar kind of relationship across. We're all anchored to the torso. Okay, so we've got a lot more movement though that can happen, and we can mess with the spatial relations a little more. So here we've got bodies can move around. We can look at how does the hand or the arm relate to the torso, what's the positioning of the legs with respect to the rest of the body. So we can again move and manipulate the spatial relations among features of a whole body, not just within a face. And I always like to put a little bit of information in here before I get into some of the interesting research data about why I even study infants in general. I had a pretty interesting path to stumbling into developmental psychology, I think. So I don't know, many of you changed my major multiple times during my undergraduate degree. Wasn't quite sure exactly where I wanted to go, but I was able to work close with one of the faculty members, and they kind of just made me understand how cool psychology is and how it's not just the clinical aspect of psychology, but how important research is to the field too. And I fell in love with research from that point on and wanted to continue and do that, follow my kind of passion there for science, and was able to apply to grad school, go to grad school. And I didn't have any experience with babies, with developmental psychology before that. I was just doing some basic research with undergraduates and stumbled into an infant development lab and just absolutely loved it, fell in love with it. I can't imagine myself studying anything else because as a developmental psychologist, I'm able to ask these kind of questions. So I'm able to ask research questions about what's the nature of processing. So those include things like, how do we process different kinds of information? So we can ask questions about emotion, facial features, relations among those facial features. We can even get into some questions about, are there dedicated facial or brain regions dedicated to face processing, body processing, emotion processing, and how does that contribute if we see differences there across individuals? So we can investigate all those kinds of questions within infancy research. We can also investigate what are the origins and potentially some of the mechanisms of development. So like I said, we all know that adults can identify different individuals, they can process emotion, they're experts at face processing, and that's cool, but when does that start? Right? Don't we want to know, when does this all begin? Maybe it's just me because I'm a developmental psychologist, but that's what I want to answer. I want to know, when does this begin? How do we actually learn about this? What does that learning look like? So by investigating different origins and mechanisms of development, I can try to ask those questions. So what components are present, what abilities are present at birth or shortly after birth with very little experience in the world? What sort of concepts are developed over time or learned from our experiences? And then the mechanism question is, well, what's actually driving that development? What is driving our learning across those different samples? So trying to ask different questions like that. So hopefully you'll see through the talk that I'm slowly working through answering some of these questions about infants. So unlike the research I was doing with undergraduates, unlike research with adults, I can't bring babies in to a research lab or go into a family's home to study a baby and give them a set of questionnaires to fill out. I could do that for the parents, right? You can definitely do that. But the baby can't do that, especially, we're nonverbal subjects. So how do you even go about doing research with babies? Hey, that was step one for me to discover when I was, entering into this field. And you just have to get creative. There's some really cool methodologies out there that you can use that help you make inferences about what infants are remembering, what they're understanding, what their preferences are. And so being able to use those methods, I think, is super cool. It's a little bit more unique. You have to be a little creative and think outside the box when you want to ask the infants these different questions. So that's what we get to do, and I'll talk you through a little bit of the main methodology that I use in my research. Okay, and that's called the novelty preference visual recognition task. It has several different names. You'll kind of refer to it across a few of those different paradigms. But for the most part, what we're looking at with this novelty preference, in very simplistic terms, has to do with the idea that infants like new things. They like novelty. They're attracted to novelty. So an easy way, if you guys don't have kids and haven't been able to experience this yourself, maybe you have an animal in your life, maybe a dog. Okay, dogs love novelty, right? Very similar. So if you bring them in, maybe you bring them a new toy. They're going to forget about everything else that's kind of around them at that time, and they're going to say, "Oh my gosh, a new toy! I want to go play with that," and that's going to get a lot of their attention. Okay, so it's this idea of that novelty preference, this attraction to new things that maybe they haven't seen for a while or haven't seen before, that can drive preferences, and in our case, it's going to drive their looking behavior. Okay, so it's called a visual recognition task sometimes too, because we're using the infant's eyes to determine what they're attending to, where they're paying attention, what their preferences are. So based on where they're looking or which picture they're looking to, we can make inferences about what they're remembering. So an example is kind of set up here. This is an old school, before computers. Some researchers still prefer these models. Okay, but you can see there's a little hole where there's a camera in the middle there between two pictures up on the screen. And that camera, or it could be a live person, is just watching to see when these two pictures are presented to the baby, which one are they looking to longer? They just total up kind of the amount of looking to one side versus the other. So I had the privilege of having my own research subject not too long ago. And so I was actually able to put her through some of these procedures. So I've got some pictures and clips to show you of that too. But I guess first, I got ahead of myself a little. The novelty preference procedure, right? We're going to put you guys through a little test. I'm going to make you work as you came here today. Okay, so imagine we're using this kind of familiarization, habituation idea. In general, that means if I show you something repeatedly over and over again, you're going to learn what that is. You're going to become very familiar. You're going to be able to study it, learn everything you can about it. And then when I show you something new following that, hey, I'm going to measure and see how you respond to that difference. Now you see two contrasting stimuli. What do you do? Okay, so you guys are my test subjects here. I'm showing you over and over again this very simple stimuli. I've got the same thing on both sides of the screen. You can look at whatever you want. You're going to get the same information. Okay, so you stare at these two green squares over and over again until you get super bored. Okay, you're welcome. Okay, and then we'll go into test trials. This is where I get the information that I want, right? So I want to see what happens now when you see something new or novel. And we're saying it's new or novel because it's different from what you've seen before. So now your test trial is going to look like this. You see your familiar green square, and now it's paired with a new novel red square. Okay, so we manipulate what's different. I could have done a test trial here that was a green circle, and that would still be a difference, right? How long are you looking? We're changing the shape that you're seeing instead of the color. So we can change different things like that depending on our research question. So we'll have a test trial that looks like this. And then just to make sure you don't have some sort of strange bias going on, maybe you like the left side over the right side, we'll switch sides of the screen that it's presented on and make sure you're kind of following that stimuli as it moves to a new position. And then what we'll do is take your total looking preference, how long did you look to the red square versus the green square across my 2 test trials, and get a nice proportion. Okay? And that's your novelty preference in the end. How long are you looking to our new novel image? So here's my awesome research subject. Gosh, she was only about 2 months old in this picture, I think, so she's younger than the age sample I study. But what we're doing for this methodology is that a parent will come in and simply hold their infant on their lap. They're in front of a computer screen that's got a camera secured to the top of it, and they're just watching images come across the computer screen, and the camera's recording where the infant's eyes are looking. Okay, so it's non-invasive. The infants usually love it. Okay, they get this extra visual stimulation. They like to look at the pictures. Sometimes there's videos. So they have a really good time with it. And I think the parents really enjoy it too, because they get to see their infant in a new environment, enjoying some sort of a task, and they feel like they're contributing and really being successful and doing a great job. The smartest babies that I've ever run, right? That's what you always say. How'd my baby do? Oh, it's the best I've ever seen, right? I always have to. And then what we kind of do on the back side is then look at the performance, so then we can use different programs to analyze, and helps us extract more and more information about it. Okay, so this is another example. I threw this in here because I think it's important to note that the parents actually can't see the pictures while the studies are happening. We actually put really technology advanced glasses on the parents, aka sunglasses with duct tape over the lenses, right? Because they're holding, my research participants, because they're holding the babies, I don't want their ability to tell the difference between these 2 pictures to influence the baby's behavior. So as much as you tell them, don't point to the screen, try not to bias them one side or the other, very subtle, unconscious things like turning your body a little could be influencing the baby's behavior. So we want to make sure we're clear of anything like that, so the parents can't even see the pictures during the actual study, just to make sure that we're not getting any sort of influence there. So they get to wear those really stylish sunglasses. And I've got a sample of what this looks like on our end of a baby participating. So imagine, okay, this is kind of when it starts, it's going to be the intro to the video. It's kind of an attention getter on the screen. You'll see the color change. So this is the first trial. The baby's eyes are looking back and forth at the 2 pictures. And what we'll do after the study's done is we'll sum how long they're looking. So we just, total up how long they're looking, one side versus the other. But it's pretty amazing to see live. You can see that they are looking back and forth, comparing the 2 pictures, and deciding which one is capturing their attention a little bit more. Which one are they drawn to and they want to look at longer. Okay? So that's where we're going to get our novelty preference from, is kind of measuring their eye movement in something similar to what you just saw there. Another time for some audience interaction. I stare at these pictures so much that this stuff looks so obvious to me. So I always like to get a feel for what you adults, you expert face processors think. Okay, so we've got 2 pictures of a face on the screen. Are they the same or different? Different. What's different about them? Spatial. You guys have been paying attention. Okay, all right, I kind of set you up for that. Very good. So yeah, we've got some different spatial relations going on between these faces. Otherwise, they're identical. So we've gone in and manipulated, okay, this face. We've changed the distance between the eyes and the distance between the nose and the mouth. They're very subtle. Okay, so according to the changes we've made, it would be just as probable, it would be perfectly reasonable for you to run into this individual on the street. We haven't changed the distance between the eyes or the nose or mouth to an amount that's, unusual or would be unlikely for you to encounter in a person. Okay, so they're all within the physiognomic norms of a female's face. Same thing for this one. This is someone you could run into on the street. So you guys did great. You notice those differences. Okay, you guys are expert processors. Way to go. Okay, and so what we're trying to see is, when does this develop, right? That's one of my big questions as an infant developmental researcher, is when do we start to be able to do this? Okay, and so I put this on here in case you guys couldn't see the differences. This makes it a little bit more obvious. Okay, So, right, that looks really extreme when they're kind of placed on top of each other, unlike when you kind of had to look at them a little close, maybe squint one eye to see the difference on the other slide. This makes it really obvious. So these are the same faces, just, superimposed on top of one another. So you get the idea. So through several different research studies that we've done over the years, we've come to find out that actually by around 3.5 months of life, 3.5 months of life is all, infants can process and are sensitive to those spatial relations within a face. So just like you guys did here, even 3.5-month-olds will be able to tell me that those two faces are different using that novelty preference procedure. Okay? So again, we see this is kind of a normal, typical face, the spatial changes that you guys already saw. So we see that only takes 3.5 months of exposure with faces or experience to faces to be able to do something that is a mark of expert or adult-like processing. Okay? This is groundbreaking, or it was within my little field of infant researchers that study facial processing, because it was thought for a really long time that it took months and months and months, potentially even years sometimes, of experience with faces before you could exhibit some of this expert-like processing. Okay, so it's really cool that we were able to show, with a few different tweaks along the way, that about 3.5 months, they're able to perform and can tell the difference between two faces with very subtle spatial spacing relation changes. I told you we'll also do something with bodies, right? So body processing sets up kind of the same. We've got spatial relations. So what I'm showing you here in this top image is what we show babies to start. So we give them about 30 seconds of experience or exposure to those stimuli. This is comparable to like what I showed you guys with the green squares. We're going to show them the stimuli till they've accumulated some experience with it. They're going to learn about that body posture, the spatial relation among features there of the arm to the torso, the leg to the torso, okay? And then at test, we're going to show them that same body posture that we showed them during familiarization with a new one where we've changed the position of one arm and one leg, okay? And we're trying to see, is that something infants are sensitive to? Can they tell the difference between those body postures? Now, it seems, again, super obvious to us because we see bodies in all sorts of different organizations all the time. So yes, of course they should be able to see the difference, but no one had asked that question with infants yet. Okay, when it comes to body processing, again, it was thought that it's not till 12 months, 15, 18 months that they show any sort of ability to be sensitive to or perception to be sensitive to bodies in general. Okay, all the research had focused on faces and the whole person was really being left out of the picture. Okay, so it's how often do you guys walk down the street and just see a headless person? Not very often. So if the argument is that we're experts at facial processing because of experience and exposure, well, we should be pretty good at processing bodies too, because we usually see a head attached to a body, right? So it makes sense that we're seeing similar trends when it comes to body processing, that infants as young as 5 months of age, okay, are able to tell the difference and look longer to the new or novel body posture when presented in a fashion like this. We also were able to do a kind of continuation of this finding with 5-month-olds and found that not only are they sensitive to these changes in whole bodies here, like you see, but they actually do this, they're actually applying holistic processing to this differentiation. Okay, so that means that they're using the body as a whole to help drive their discrimination between these two pictures, or how they're telling the difference between these two. So if we just showed them individual body parts, like the arm or the leg that are changing, removing some of the extra information that stays the same, they actually don't look any different, or they don't look any longer at the novel relation. They actually need the context of the whole body to be able to do it. So that's really cool, and that again follows what we know from the facial recognition literature, that it's a holistic process, not the individual parts that are actually driving their performance. Okay, so it's really cool. We're seeing kind of parallels between the face literature and this emerging area of body processing. All right, once again, I have a little bit of kind of audience interaction here to keep you guys awake. Okay, so we're gonna play a video, or 2 videos are gonna play on the screen here side by side. Now this is a slightly different methodology, so it's gonna make you guys guess a little bit more. What I want you to try to guess, or tell me what you think will happen, or what an infant would do, is are they gonna look to the video that matches the sound that I play? Are they gonna look to a video that doesn't match? Okay, which one do you think would capture their attention more? Okay, so let's see if this works. So not only is that creepy, okay, but it's fun too. So what was the vocalization? What was the sound you guys heard? Can you guys identify it? If you had to label it of some sort, what would you label it as? Laughing. Oh, gosh, you guys are great. Yeah, so it's laughing. It's a happy vocalization. Okay, so it's playing along. So which video did you guys find yourself looking to longer during that? The one on the right? Okay, so the one over here, I got a vote over here. And what was that video doing? They were kind of dancing around. Okay, that's a happy video. It was supposed to be, an actor displaying happiness. Okay, and on the other side of the screen, it was paired with an actor who was mad, angry, right? They're kind of stomping around, being grumpy. Yes, it was an anger video. So we're looking to see if presented with two different emotions and a vocalization that matches one, what do infants do? Okay, and what we actually found, to not really our surprise, this was in direction with what we thought, but infants were sensitive to emotion in bodies because they demonstrated that they look to the body that matches what they're hearing. So if they hear a happy sound, they look longer to the happy video. And we also did it if they hear an angry sound, those infants in the angry condition looked longer to the angry video. So the only thing that differed between our conditions there was what they were hearing. And so depending on the emotion that they were hearing presented, that changed what video they were looking to. So infants actually match their auditory, so what they're hearing to what they're then choosing to look to. They show this really nice matching preference across development. And that lets us know that they're sensitive to emotions in bodies. So not just can they do these spatial relations, but they can use the spatial relations and some of the inference they know about even emotions, and they're sensitive to emotions in bodies. The facial emotion literature's been around for decades and decades. Okay, so we know that they're sensitive to emotions in faces, but this body idea is again new. So we're finally able to demonstrate infants can pick up on emotions in bodies too, not just faces. So this work shows, there's different renditions of this. So broadly, we see it begin at about 3.5 months. We see this sensitivity to emotions in bodies show up a little bit, and then we see it just gets kind of improved upon, it gets stronger, it kind of takes different forms, gets a little bit better over time. So more experience, they become better, can do it in, tougher conditions. Okay, so by 5, they get a little better. By 6.5 months, it's improving and improving, they can do it for more and more emotions. They can do it across different types of stimuli. Okay, so we see that change. But in general, within the first 6 months, within the first 3 months of life, we do see they're sensitive to emotions in bodies too. So I told you a little bit about the emotion in faces. What we're trying to do here is understand a little bit more about what the attentional processes look like. So instead of just looking or knowing how long infants are looking to a certain stimulus, so are they looking to the novel stimulus or the familiar stimulus? In honesty, we got a grant, so we got some money, and we're able to buy a fancy camera. Okay, it's an eye tracker, and what it does is it not only tells you how long they're looking to one stimulus versus the other, but it actually gives you a lot more detail about where they're looking on the picture. So not just broadly, I'm looking to the left or the right, say I'm looking to this left eyeball, or I'm looking to the right hand. Okay, so we get a lot more detailed information about where infants are looking. So with that technology, we can ask some different types of questions. We can use some different methods to continue to explore the same topics of facial processing, body processing, etc. So what we started out doing was to investigate facial emotion. We wanted to take something that was pretty consistent and well established in the literature and see if we could replicate and extend it using this really cool new tool that we had. We wanted to make sure we knew what we were doing. So we wanted to see if infants look differently or their attention is distributed differently across emotions. Okay, so in this clip, we've got, I know this is kind of small on there on the screen here, but we've got an actress in the middle who displays 3 different facial expressions. First one is happy, so she goes from a neutral expression to happy, and she does a kind of fearful expression, so she looks afraid, and then the last one she stays neutral. She just keeps a neutral expression throughout the entire clip, but she blinks. So it's still considered, a video and not just a still image of something. So it's supposed to help the infants, pay attention a little bit more, help increase the ecological validity. It looks more realistic, right? They're always encountering moving faces in their world, so it's supposed to kind of help. And then what we're looking at, I'll see if I can get it to maybe play again. You saw this cool little red line jet out, right, from the face over to the side when this checkerboard popped up. And what we're measuring is how long are they spending on the face in the presence of that checkerboard, right? It's really distracting. Babies love high contrast. They like things that flash. Even us, when we look at that clip, when something new appears, it's really hard for us to can maintain our attention on there. We really want to jump over and see what just appeared. Okay, so infants are the same way. So how much are they able to inhibit that response and stay on the face? Or how long does it take them to reach that? And does that vary depending on what the facial expressions are? Now, we chose the emotions that we did here because fear is considered something that's really special when it comes to emotions. Fear is just a little different than the other emotions, much like happy is sometimes considered different because they evoke such strong responses from adults and infants alike. So fear is unique because it's evolutionarily important for us to be able to detect this and recognize this really quickly, right? If someone else is responding, is afraid of some sort of situation or some sort of stimuli in their environment, that's important for us to be able to pick up on and then respond accordingly too. Okay, if there's something dangerous in our environment, we need to respond and change our behavior, get away from it, flee a little bit. So being able to detect fear in your environment is critical for one's survival. So we think fear is going to be a little different than happy and then the neutral expression. And that's actually what we were able to find here within our group of 5-month-olds. So this is actually one of the first developmental differences I've presented to you, where we've tested infants of different ages, so 3.5 and 5 months here, and we actually found a developmental change. So this is something that wasn't present in the infants we tested at 3.5 months, but within the 1.5 months, of extra experience, exposure in the world, something is changing, and by 5 months, they are sensitive and they're treating fear differently, as evidenced by the differences in our groups here. So this darkest bar here is fear, followed by happy, followed by neutral. And what we're measuring here is how long they're looking at that face when that distractor is up on the screen too. So how much are they still attending to that emotional face? And they keep their attention on that fear face. It maintains their attention longer than happy and neutral expressions. So they're trying to extract that. It's different from the other emotions. So we're able to replicate the fear bias that we've seen in the literature and show that infants as young as 5 months of age show this sort of fear bias when it comes to attentional processing. And so this was an extension of that previous work. Previously, it was thought about 7 months. So, that's not anything that's super groundbreaking, right? We're not going out writing something new here for the field. But one of the goals that I have as a developmental researcher, as an infant researcher, is to investigate those origins of development. So if we show that something is present by 7 months, well, what can we do? How could we see if it's present at a younger age? Okay, so we added the videos. That's more realistic to what they're seeing. Maybe that helps them show us that, yeah, I can do this, right? So now what we're continuing to do is investigate why our 3-month-olds aren't doing this. What's different? So now we're exploring that mechanism of action. What's driving changes between 3.5 and 5 months of age? What's helping, what can be going on to explain why my 3.5-month-olds are failing, but my 5-month-olds are succeeding? We want to try to understand that change a little bit more. So that's where we're kind of working towards now. So to briefly summarize some of the studies that we've talked about already today, is that we see that by 6 months of age, in a lot of cases, infants are sensitive to these really subtle spatial relations in faces, the spatial relations in bodies. And we also see some evidence of an ability to perceive emotions in faces and bodies. So not only, we've covered a lot of areas of social perception, whether it be through face processing, body processing, and emotion processing in both of those modalities. So what I will finish my presentation today on is talking about some of my research on opiate-exposed newborn infants and what we know about opiates' influence on their early cognitive development. Here in Tennessee, so this was the most updated piece of information I could find. They're a little behind because this only goes through August, about the rates of NAS, or Neonatal Abstinence Syndrome. And so this coincides with opiate exposure because a good percentage, okay, so the percentages actually vary, but around half to up to maybe 90% of infants who are opiate exposed during the prenatal period go on then to develop a clinical diagnosis of NAS, or what is now referred to as neonatal opioid withdrawal syndrome, which is abbreviated NOWS. So we'll kind of go back and forth between NAS and NOWS because not a ton of people can recognize NOWS as NAS. So trying to introduce that new term and weave it into, but that's what we're, the updated and more accurate term for what these babies are going through is. So NAS or NOWS is really important to understand during infancy because it leads to some pretty uncomfortable symptoms for the baby. So when they've been exposed to opiates, opiates actually cross the placenta during development and can then impact fetal development, brain development, a couple other areas along those lines. And we want to try to understand what impact that has later on. So in the newborn, once they're born, what about, shortly after birth? What sort of impact is that having? What about as they're entering preschool? What about when they're starting to enter schools? Are we seeing differences between non-exposed, non-opiate exposed peers? Or is their performance really not impacted by their prenatal opiate exposure? And again, I have the previous slide on here because this is a big problem in the region and location that we're in right now. It was also a problem in Kentucky when I got started on this, but this is a big reason why I wanted to come to ETSU, is so I could continue to work with this sample because you guys are right in the heart of the opiate epidemic here. Okay, so trying to address and understand what sometimes are the forgotten kind of component of the opiate epidemic, these babies born to moms who have used opiates. So we've got NAS kind of going through, it's related to several withdrawal symptoms that aren't fun for them to go through when oftentimes they're admitted to the NICU to help with those withdrawal symptoms, which increases their length of stay at the hospital, increases the cost to the hospital to care for them, and is related to a couple other adverse kind of birth outcomes as well. Developmentally, we know a little bit more about some outcomes associated with cognitive development thanks to our colleagues that study animal models, so specifically to various rat models, because you can manipulate drug exposure and learn a little bit more about the impact there. So animal models have helped show us that that opiate exposure impacts their attention and memory processing, specifically spatial information. Right, hence kind of the theme for the talk here is that when these animals are placed in a maze, they actually show impaired memory for how to kind of complete that maze, right? They forget how to kind of manipulate that. It takes them a little bit longer. Okay, so there's something impacted within, or the opioids are then changing their memory development, their attention development, their spatial relations. There's something potentially going on there that we can take the information from the animal models and try to then study in human infants. So that's what I was trying to go after here. And so what we did is investigated whether there are any differences. We don't know still, right? We're still in an investigative stage. We're trying to understand and gather information about whether there are differences between opiate exposed and non-opiate exposed newborn infants when it comes to their spatial processing of faces and other objects. So that's what this investigation was trying to do. This says 96, but the data I'm presenting is actually just from 64 of those infants. So we ran 64 infants using that same procedure I showed you guys earlier, that familiarization idea, looking for this novelty preference over time. These stimuli look a little different from what you saw before, and this is because for newborns, we actually have to remove some of the distracting information from a face for them to be able to succeed on this. So with typical developing infants, there's research showing that they can process these spatial relations in faces if you remove the hair, if you remove the ears, and the neck information. Because otherwise, they'll just use that external information to try to identify differences between the pictures. It's a little bit easier for them. It's a little harder for newborns to process these really small, subtle facial features, when they have something else that's easier for them. So we remove the easier stuff and really force them to pay attention to those internal features, then we can see that they do process different. They can tell the difference between those two pictures, between the spatial information. So again, they're gonna see a 30-second familiarization, they see a cool attention getter come up, and then they see test trials. Okay, so again, you've got the same face they saw during familiarization paired with a novel face, attention getter again, and then they switch sides. Okay, so very familiar, should be a familiar procedure to you all now. We also ran these infants through a task that looks at spatial relations that aren't in faces, so something that's not a social stimuli, maybe something that doesn't have that social kind of priority that would be indicative of expertise, or they have this extra attention to be drawn to. Faces are just kind of special, right? So we wanted to give them an option or see if their performance would differ on just basic shapes. Okay, so here we're looking at spatial relations in a vertical bar in a box. Okay, so they're familiarized to this spatial relation and then tested with a familiar versus a novel spatial relation. Okay, nothing too crazy exciting there, but it gives us a comparison point. And again, then they change positions. So usually people always ask, most people don't know that, or still think that infants are blind when they're born, right? This was put forth by a couple pediatricians several years ago, and so it still makes its way around. But in fact, infants can see shortly after birth. Their acuity is just much more poor than ours, or our acuity as adults. So these figures kind of try to help you see what it maybe would look like through the eyes of an infant. Okay, so this is kind of what we see, and again, I understand these figures are kind of small, I apologize. This is what we see with the clarity, okay, and at about 30 centimeters, which is the distance that we're showing our pictures to these newborns, okay, that's what those differences would look like. So it's blurry, okay, for a lack of better words. It's a little blurry. You can still make out there's some differences. Okay, if you look hard enough, you're a little bit closer than you guys are now, you could still tell that there's different facial expressions on those different individuals. Okay, so it's definitely not as clear as what you and I see here today, but infants are able to tell the difference between different facial expressions, even with their decreased visual acuity at birth. This is a little bit of information about what my participants looked like within my sample. Okay, so my newborns, those are hours old. So within my sample of non-opiate exposed infants, they're about 37 hours old. Okay, so this is the youngest sample I had ever worked with. When I was recruiting them, they could be born as young as 12 hours old, all the way up until 72 hours old, because that's when most families are discharged from the hospital if there's no other complications that they're trying to keep an eye on. So they were anywhere from 12 hours to 72 hours old, averaging about a day and a half. Okay? Crazy amount of different exposure than my 3.5-month-olds. So less experience going on here. And imagine how much they're probably sleeping for those 37 hours of life, right? Newborns sleep all the time. And then my opiate-exposed infants were about 173 hours old, so they're much older, but the opiate-exposed infants are in the hospital longer because they're being monitored for symptoms of withdrawal due to the prenatal opiate exposure. So if they've got a diagnosis of NOWS or NAS, okay, they're being monitored and potentially treated with different drugs to help lessen the symptoms of their withdrawal. So that makes them stay in the hospital a little bit longer. So they just, out of opportunity, could be a little bit older. Okay? It gives you a little bit of information about some of the other outcomes. Not a ton of differences. The head circumference of the opiate-exposed group was a little bit smaller. That's got some implications for cognitive development down the road too, so we want to make sure we're accounting for that. But really, for the most part, nothing else is going on differences between the two groups when it comes to birth outcomes. For me, this is a little bit more information about mom and what her information looks like. So this is kind of SES information, or education levels. So as is predicted by several other studies, we do see significant differences in the education level, the age, and employment between moms of opiate-exposed and non-opiate-exposed infants. So what I did is I ran my babies through the study looking at facial expression. So they did the face study, then they did my non-face or my shape study, and I am plotting here for you guys what their visual preferences looked like. Okay, so what I have here for reference is a bar right at 50%. So with novelty preference procedures, right, if you're looking to both pictures evenly, okay, your novelty preference is going to be about 50. So anytime that we see infants looking performance significantly differ from 50%, okay, that's letting us know that they're looking to one image significantly longer than the other image. So we see our non-exposed groups here in my blue bar all the way up probably around 63%. Okay, and statistically that is different than 50%. So that lets me know my non-exposed infants are telling the difference between those two facial images. So they're sensitive to that subtle spatial information within the faces. That's what we kind of predicted. That's great. Okay, so we see this. In my non-exposed group for the non-faces, same thing. They're significantly above chance. They're showing a nice novelty preference for that new novel spatial relation in shapes. Now in the red bars, here's my opiate-exposed group. and they're almost right at 50%, okay, for my face condition. So this is showing that they're really not having a preference for one over the other. They're still looking, they're attending, but they're looking to both faces equally. So neither one is really popping out as different. Okay, or if it is, they need to we have to explore that a little bit more. It's not within the same experimental conditions as the non-exposed. So we see their performance is no different from 50, and the same thing is supported in the SHAPE study as well. Performance is no different. They're not really attending to the novel or the familiar image longer in that condition. So within the model that I ended up running, I'm controlling for some of those significant differences. I pointed out in the table. So the age, it could just be my non-opiate exposed infants are younger, and at that age, they do this. When they're a little bit older, they don't. Okay, we could see some weird trends. So when you control for age, when I control for those SES differences, and when I control for that difference in head circumference, statistically, okay, that's showing me that I still get this really nice group difference. So overall, we're seeing that, okay, across my stimulus conditions, my face condition, my shape condition, that performance is different between my opiate exposed and my non-opiate exposed group of infants. Okay, controlling for those differences. So that's letting me know that there is an association, okay, between opiate exposure and their performance on these two specific spatial processing tasks. Okay, you'll notice, for any of my child psych intro students who may be here, what I did not say is that opiate exposure causes differences on spatial processing tasks, right? I'm not doing an experiment, okay? I can't manipulate opiate exposure. So the best conclusion I can draw from this data is whether there's some sort of relationship or association, okay? So that's something that I wanna drive home, that you guys understand, is that it's not saying or implying some sort of causation. We're talking about just an association. And again, that's because my data are correlational in nature. Okay, we also had some difficulty matching groups, which you guys saw, but with my differences there. Ideally, I would have been able to control for age and match that in my samples, could have matched for SES in my samples and some other types of exposure, but we weren't able to do that. But that's kind of what's headed in the future. That's where we're headed, is trying to have better and more controlled matching across my groups. And then I had a lot of variation within my opiate exposed sample. So it's just opiate exposed in general. Some of these babies had a diagnosis of NOWS, some of the babies did not, okay? And what we really need to try to tease apart in the future is if there's a difference between those two subgroups. So is there a difference between infants who are opiate exposed and then go on to get that clinical diagnosis of NOWS, and those infants who are opiate exposed and do not go on to receive that diagnosis of NOWS? Okay, so both of those are opiate exposed, but different diagnosis, different treatment within the hospital stay. So are there differences there? And compare that to, again, a healthy control. Additionally, we'd love to be able to follow these infants longitudinally. Okay, right now we're just staying within the hospital setting, but we'd like to be able to follow them over time and see if these differences remain that we're seeing. Are there changes? Do we see these group differences increase? Do they decrease? Okay, we just want to investigate because we don't know right now. Okay, this field is wide open for more research because we just need to answer all these questions. There's so much that's unknown and we want to learn more and more about. So where I'm kind of headed, right? We're talked about longitudinal research, trying to disentangle, right, those effects. So this is trying to understand differences between opiate exposure versus actual diagnosis with NAS. And what I'm currently investigating is this question right here. So is there a correlation of this novelty preference of this early cognitive development that I'm finding here? Does this relate to their motor development? Does it relate to some other type of development that we can measure early in life to see if it extends to another kind of domain of development? Okay, and so my colleague and I received an RDC grant from ETSU to do just this. And so we are now IRB approved and we'll be starting data collection in January to investigate that question here at JCMC. So I'm very excited about that opportunity. And eventually down the road too, we want to try to tackle some of these questions about other types of social competence. So do they process information about different genders? So they process male stimuli the same as female stimuli? Do they process differences across other aspects of social processing. And then again, we've got emotion. So doing some of those emotion studies I showed you with those cool, creepy actors. Okay, what do the newborns do? And are there group differences for some of those emotion processing questions too? So that's where I'm headed, hopefully in the future. Give a quick shout out to all of my collaborators who helped with a lot of the data collection that you saw presented here, and the other funding agencies that helped support this research through the years. And yeah, finally, just thank you guys for your attention here today. And I kind of want to open the floor up to any questions you may have. Thanks. Yeah.
So I was wondering if you noticed there was a difference in gestational period of opiate use and of cognitive development, like if there was if the infant or the fetus was exposed to opiate use in like the first or second trimester versus later on.
That is a great question, and it's something that we hope to be able to document better in the future. But within our sample here, we weren't able to get information like that from the moms. We just knew if there was a history in general. But absolutely, there is some research that's starting to show the timing of the exposure is very important to sort of the impact it has on development. So being able to monitor that and incorporate that within our research is a direction we want to move to in the future. Yeah.
Would it be possible to get like dosage or what type of opiates from the moms?
Absolutely. So I do have some information about the types of opiates, and in certain situations we can get some information about dosaging, especially if they're on MATs, so medically assisted treatment. So those all come with various dosages. So we can get some information on that to incorporate too. But again, my sample was so variable that being able to look at that statistically just wasn't an option. I've got to get a much bigger sample, and we're actually going to be able to look at some things like that through a large chart review that we've actually put together in conjunction with the Department of Pediatrics to look at all births at the local hospitals around between 2011 and 2016. So this is a great sample, a great resource. It's got around 460 opiate-exposed infants, and we've got matched controls. And we're going to try to answer or ask those specific questions. Are there differences based on dosing and the type of opiate they were exposed to? Mm-hmm. All right, we're almost at 1 o'clock. Any other questions? Okay, well, thank you guys for your attention today. Have a wonderful rest of your Wednesday. And I'll stick around if you want to ask anything too.